This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 236, recorded on February 11, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
My pleasure. Thank you.
Elio, you got vaccinated, right?
I sure did.
You got two. Wonderful. Excellent. Yeah. Glad to hear it.
No reaction to the second
Beautiful.
one, which worries me.
No, you're fine.
It worries me.
That's fine. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. Yeah, it's the snowy North Coast. We've got probably 6 inches of snow.
Hey, we have a foot here.
Woo.
Yeah, we've had now 3 snowstorms in the last 3 weeks, and it's really cold here, so it's not melting. Michael Schmidt will join us shortly, we hope. I told Michael, he said, I'll be late. And I said, well, if you don't show up, we'll be done in 15 minutes. Was that a nasty comment?
Unfortunately, it's true.
We love Michael. Yes, Michael likes to chat. Okay, today we have a snippet first from Michele, something different, Michele, right?
Yeah, the article is entitled The Academic Career Readiness Assessment, Clarifying Hiring and Training Expectations for Future Biomedical Life Sciences Faculty. The authors are Lawrence Clement, Jenny Dorman, and Richard McGee. They're from UCSF and the Feinberg School of Medicine at Northwestern. This was published in June of 2020 in Life Sciences Education, which is a journal of the American Society for Cell Biology, and it's open access. And it was a project funded by UCSF's Office of Career and Professional Development and also the Burroughs Wellcome Fund Career Guidance for Trainees Award. And their product earned the award from the AAMC, and I'll have to locate the name of that award. But here's the story. Their project goals were to advance equity in mentoring, transparency in hiring, and accountability in training of aspiring faculty in the biomedical sciences. So why do we have a need for this? There's actually a strong literature showing that many trainees depend nearly exclusively on their mentor for career guidance. So that's flattering, but it's also risky for many reasons. So first of all, by definition, their mentor only has experience in their own chosen career path. They're not going to be familiar with other types of academic jobs or other jobs out in industry or with agencies, for example. Moreover, there's research, including by Dr. Kenneth Gibbs, who's an immunologist who's now at the NIH, and his portfolio there includes undergraduate and pre-doc training programs and the Mosaic K99 and other programs that are just designed to recruit and train people from our full pool of talent in the biomedical sciences. And what Gibbs and others have shown is that mentors represent, unfortunately, a significant source of bias, whether it's conscious or unconscious, in who they encourage to follow what career path. So this creates a barrier for trainees who don't fit the stereotypical faculty profile. So if they're not a privileged white male, for example, with an extensive faculty network, they may not be encouraging trainees. Well, it's clear they're not encouraging trainees from underrepresented minorities or people with disabilities, what have you. There's also literature that shows there is bias in letters of recommendation written for men versus women, and also disparities in letters written for our trainees from underrepresented minority backgrounds. So this project has set out to address these and other challenges in our field. So the people that can benefit are aspiring future faculty, so all our trainees out there who are thinking that they want to follow an academic career path. Also the mentors of our master's students, our pre-docs, and our postdoc trainees. Also, our career and professional development teams at our institutions that are working hard to provide transferable skills to our trainees so that they're ready for the competitive position of their dreams. And then, importantly, search committees for faculty positions can also benefit from understanding the literature and putting practices in place that minimize bias. So what's really great about this tool is it is directed at 3 different types of colleges and universities that we have in this country. So the research-intensive universities, or R1 universities, where the primary faculty responsibility is research with some classroom teaching, but mostly mentoring trainees in the research lab. It will also help people who aspire to jobs at teaching-only colleges, like our community colleges, as well as the third class, where both research and teaching are a focus. So these include, for example, our liberal arts colleges, or what we call primarily undergrad institutions. So how do they do this? How do they address these lofty goals? They have developed a rubric by doing not only careful literature research, but they also had intense interviews with people from 20 different educational institutions who themselves had multiple experiences on hiring committees for faculty positions. So they use many rigorous scholarly techniques to come up with this vetted tool. So how does it work? It actually can function at 5 different levels. It can provide aspiring faculty candidates transparent criteria so that they better understand what the expectations are of somebody in the path that they want to follow. It also will allow the trainees to identify early on in their training what opportunities they need to seek so that they will be competitive when it's time to go on the job market. Also, it will provide trainees with a vetted structure where they can use to get feedback from their mentor, so that they can have structured discussions, and their mentor can better advise them in ways they can get the skills and competencies that are going to make them competitive for the path that they choose. And then also it can standardize the evaluation process of faculty candidates. So this addresses a common bias in our search committees, where it's easy for the members of the search committee to kind of slip into the mode of, I know talent when I see it, we're just looking for a good fit. And again, literature shows that there are better practices that can reduce bias. And these include having the search committee members get together before they've even posted an ad and discuss what are the qualifications that we know are required for success in our department at our institution. And also to make a list of those and be actually deliberate about evaluating each candidate in multiple categories rather than just taking a stack of applicants and choosing their favorite one. So by eliminating this kind of inherent bias of like good fit, search committees can be much more intentional and deliberative and look at the full portfolio of skills that are needed to be successful at their department and their institution. And then finally, it can evaluate people that are running training programs or career and professional development offices so that there is a rubric that funding agencies can use to evaluate whether they are using best practices to provide all of their trainees the necessary preparation so they'll be competitive for whichever faculty track they may choose to go. So clearly this is very ambitious, and what does it actually look like? It simplifies to a table that lists 15 different qualifications. For example, teaching experience conducting research with students, collegiality, publications. So 15 of those qualifications. And then it has 4 different columns, level 1, level 2, level 3, and level 4. Each one describing what they would expect if you're just developing the skills, or level 4, you've mastered them. And then each of the quadrants is coded so that you can see what their analysis shows, what the expectation is at a primarily teaching institution versus a research-intensive institution. So how does this actually work? Let me give some examples. For teaching experience, at level 1, the candidate might have had a little experience as a TA, a teaching assistant. Whereas at level 4, candidate has been fully responsible for organizing and teaching a variety of courses with a comparable student population. And then underneath, they indicate in color coding and percentages what percent of the institutions in their study group said that, yes, this is an essential qualification for a successful applicant. So, for example, teaching experience, 100% of the primarily teaching institutions will require that, whereas 0% of the primarily research institutions say that that's an absolute requirement for a candidate. We also have, for example, inclusion of undergraduate research experiences in the research plan. Again, if you want to be at a institution where the faculty do both research and teaching, 100% of those institutions do require that you have some experience, including undergraduates, in your research plan. Let's look at some at the other extreme, the research intensive. For example, research independence. 100% of the research intensive universities require this, and 0% of the teaching intensive institutions require it. And what do we mean by research independence? Well, to reach level 2, for example, the candidate needs to show that they can lead a research program, develop ideas, and establish new collaborations independently. But to be at the highest level, you need to not only have those skills, but also have an indication that your research project is actually independent of your current advisor's project, so that the committee can be more sure that you're not going to be in direct competition with your PI. And then other traits like strong letters of recommendation are required at all 3 types of institutions, and collegiality is another trait that everyone is expecting. So again, in this table with 15 qualifications, 4 different levels, and then data underneath showing which types of institutions have this as an expected qualification, it means that both aspiring future faculty, their mentors, and also people who are running career and professional development offices can use this to better prepare a full portfolio of the training that somebody on that wants to follow that career path should be getting as they move through their graduate training and also their postdoctoral experience. So it can help our trainees look and see what the playbook is and decide which path they want, and then have those conversations with their mentor, with other allies at their institution, so that they can make themselves have the credentials they'll need to be competitive. And then also it is, again, for search committees, it can help them really be careful about introducing biases and just looking for the, quote, best fit, which unfortunately can exclude many people from non-traditional backgrounds. So what do you two think? You've each been on search committees and have encouraged people to go on these paths. Do you think this is going to be a useful tool?
Yes, I would say so, because to lay out the criteria on a table is a useful thing. We don't do it normally, in my experience, and so we miss out on some of these things. However, I fear that this may be a bit of a straight-jacketing exercise. That is, you have to fit in the mold that they tell you. And that may be not a problem in most cases, probably, but in some cases it may be, we have to fill that box. Let's fill that box no matter what.
Yeah, so let me be more clear on what their philosophy was going into this. They describe it as a rubric, and they've designed it.
What's a rubric, anyhow? What's a rubric?
It's a guide. It's a guide, a process that you would follow, rather than a formula that you would plug parameters into and get the answer. So, for example, by having this table, the search committee can have an open discussion before they even write their ad and assess, what are the qualities, what are the traits, the experiences that are going to help a candidate be successful. And let's rank each of our candidates in multiple categories and not just one. But the group is not suggesting that everybody use the same criteria, but rather let's be open and transparent about the qualities that our particular group is looking for, and let's be really systematic about discussing those and looking for candidates with those traits, and even putting that information in your ads, making it transparent to the candidates. They also want to recognize that there are going to be ways to improve this tool. And so one thing I was impressed by is that on their website and also in the article, they include links so that both trainees and faculty who have used the tools can go in and answer their survey so that they can continue to collect data from the field and fine-tune the rubric accordingly.
And from my experience on search committees, here anyway, they just look at your research, whether you gave a good talk, So I think having a table is useful to say, hey, there are other things you should be looking at. So I don't worry too much that it'll be a straitjacket, because I would look at them as guidelines. Just look beyond what you've always been looking at. And I think that in itself is useful, right?
Yeah, just to have that conversation at the search committee and try to reach consensus.
I think the idea that the committee should meet ahead of time and go, this is great, because usually they don't. In my experience, you just put out ads and you start interviewing people, then you have a meeting. So I think it's a good start. I'm not sure how you encourage people to do this, though. That's the problem. Every university is pretty much independent. Who's going to send this? Who's going to pay attention? And that's a bigger problem in my view.
Yeah, this is something we talk about a lot. I have a role as director of our postdoc office. So, one place that can make a difference is our funders. So, for example, at the NIGMS of NIH, they fund a lot of training grants, and they can use this rubric to go in and ask, do you have a career and professional development office that is providing opportunities for students to get these other skills? And are your search committees also using these?
I think times have changed, Vincent. In my old days, this would have been an external force. Now, I think these things are embedded much more in the process of searching for faculty and students and everything else. We're really in a different mindset now. So it would work much better now, I think. This wouldn't have had a chance 20 years ago.
Right. And that's something we can be thankful for. I think the attention that many groups and people are putting on diversity, equity, and inclusion. And we realize it's not enough just to talk the talk. We've got to walk the walk. But even well-intentioned people who want to have a more inclusive search, if they don't have the tools, they might not quite know where to start. So I think this is going to be a terrific addition to that toolbox.
Michael Schmidt has joined us. Welcome, Michael.
Hello, everyone.
And let me just say that this tool, the Academic Career Readiness Assessment, was the winner of first prize at the 2019 AAMC Innovations in Research and Research Education Award. So
good for them.
Yeah.
Do you think the AAMC could at least help the academic medical centers do this either through their accrediting activities of the AAMC to effectively encourage this sort of behavior?
Certainly. That's another leverage point. Accreditation doesn't happen all that often, but certainly.
Well, it's every 7 years for med schools.
Right.
All right. Well, I think it's a good idea. I think it's a start, right, Michele? It's going to have to go through some phases.
Yeah, and it's certainly not they're not the first to try to teach better practices to our search committees, but this is, I think, the first time that I have seen a really transparent tool that is open to all people. It's accessible online, so people who are undergraduates or graduate students, you can look now and learn more about just what do we mean by a primarily undergraduate institution and an R1 institution. What do these terms mean? What are the expectations? Where do I see myself? And how can I get there?
Got it. Thank you, Michele.
Yeah.
Elio, what do you have for us?
All right, I'm going to change gears completely. We're going to talk about the bottom of the ocean.
Love
about
it.
going
Love
from
it.
great heights to great abyssal depths. Okay, the subject is giant tubeworms. What are they? If you go down to the bottom of the ocean, by and large, you see nothing. Most of it is barren. You have a wonderful submersible gadget that takes you down. You can take pictures, you can take samples, and by and large, you see nothing. An occasional fish will come by very lazily. But then you come to a place which is full of life. And what is this place? These are hydrothermal vents. These are holes in the ground in the bottom of the ocean where what's underneath comes to the surface and makes a vent. And these are few and far between. They are at long distance from each other. But when you get near them, you find that they're full of life. And the life is not right at the place where this magma comes out or whatever it is. It's not magma, but whatever comes out, but nearby. So nearby, you have temperatures which are warmer than the rest of the ocean, about 30 degrees centigrade or something like that. And you find these giant tubeworms. These are things which are about 2 inches wide, 4 centimeters, and they are about 2 meters high. So they're long.
I couldn't believe that. That creeped me out.
Absolutely.
A 2 meter long worm.
Imagine being in the bottom of the ocean, seeing nothing for hours, and suddenly coming into this landscape. It's unbelievable. And when people first saw it, they didn't believe it. Anyhow, they got these things up from the ocean, and there's a group, several groups of people at the oceanography, major oceanography institutes are high on studying these things. So what's going on here? What are these made of? Well, if you look inside, if you look at these tubes, they're white. They look like a tube, like a plastic tube. And the very top, however, is a plume of red stuff. It's a very colorful,
feathery.
Feathery.
It's feathery. Right. It's like gossamer wings, if you will.
Exactly, and a bright red. And why are they red when the rest of the animal is white? Because it contains hemoglobin of all things.
Wow.
And so what's going on here? Well, what's the source of energy? How do these guys live? What do they live on? And so forth. And this is a whole science. And the science is made up of the following parts. If you look inside of the worm, you find that it's full of bacteria. Okay? These are bacteria that take H2S, by and large, and other such compounds, and oxidize them. And in the process of that, they create a lot of material that's useful, like it's a form of auxotrophy. It's like photosynthesis on its chemosynthesis. Okay? And how do they do it? Well, they take H2S and oxidize it with using, of all things, oxygen as the main oxidizing compound. There is, believe it or not, there's a fair amount of oxygen in the ocean. Okay, so this is not the problem. So if you dissect the tube, you take the tube up to the lab, you bring it up in your submersible, you finally made it up, you breathe the nice ocean air now, you take it to the lab in the ship, and you open it up, and so what you see is that inside the tube is a packet of bacteria. The bacteria are contained in cells called bacteriocytes, just like the bacteriocytes in symbionts of insects. These are bacteriocytes because they're cells which contain the bacteria, and they're contained in packets, in globules. Okay, now that's all very nice. Only if you look, and there's some circulations, there's some blood vessels that go through these structures. So now, if you look, this is the story, basically. The bacteria produce yield the compounds that are made by auxotrophy, by the non-light equivalent of photosynthesis. Okay. Now, if you look at the bacteria, and by the way, there are several different types that can do this. It's just not one kind of bacterium. So there are several symbiotic bacteria, which, by the way, are acquired from the environment. These are not inherited when through the eggs of the worms. They're actually acquired anew. So there are different kinds. They're all either epsilon or gamma proteobacteria. Anyhow, there's a whole collection of them. So now you look at the bacteria, and what do you see? You see that they better be heterogeneous. That is, they better be more than one kind, even though it's the same species, the same 16 sRNA. You find that they're different in function. And why do you say that? Because within each globule, each one of these packets, you find that there is more than one kind of photosynthesis, auxotrophy. Sorry, not photosynthesis. So
Chemosynthesis.
Chemosynthesis. Thank you. There's more than one kind. There's also more than one way of. Some use nitrogen as a hydrogen acceptor, electron acceptor. Others use oxygen in the same group. So what does it say? It suggests strongly that these bacteria are heterogeneous. And if you look inside each one of these globules, you find that the center of the globule is where the main blood vessel goes through. The bacteria near there are tiny, between under 5 microns in length, whereas the bacteria at the edge are huge. They're 20, 30, 50 microns in size. Okay?
Yeah,
so what's
thinking about that, I would imagine the first person that saw this population of microbes in these tube worms assumed that it was many different kinds of species, because there were rods and cocci, all different shapes. And classically, that's how we distinguish different types of bacteria based on their size and shape. But my goodness.
So this is weird. This is
really
you're absolutely right.
Weird and wonderful.
So, weird and wonderful.
And just remember, in the microbial world, surface area to mass ratios matter. They determine growth rate. And we all remember our high school geometry, 4/3 pi r cubed.
Speak for yourself, Michael.
4/3 pi r cubed will give you the surface to mass ratio. So the microbes growing that are itsy bitsy tiny ones are growing the fastest, while the big suckers at the end.
Not necessarily. That's the theory.
Okay.
In reality, that's not quite true. The big guys grow quite a bit. They grow quite a bit. The small guys, which are near the center, are apparently more involved in metabolism. And they of course, grow centrifugally, if you wish, towards the edge and become big. So what are they doing? If you look at the composition of the bacteria, the small ones are actually more involved in cell division. Not necessarily growth, but cell division. So they have things like FTSZ, the magic protein that makes the constrictions in the rod-shaped bacteria and other bacteria, and makes the septum eventually, is involved in making the septum. Those are typically of the small central cells. The large cells don't have that, and they are more involved in they have other cell division things, but they do more carbon dioxide fixing. They do more of the metabolism, if you wish. So the way they come up, I'll give you the conclusion. The conclusion is that the small cells in the middle are like stem cells. They're the cells from which the rest originate. And the big cells are the workhorses. That's where the chemical action is. So that's the story in brief. It's a magnificent story.
What I was so impressed by, and you made it sound just so elegant, is that the sophisticated methods they used to figure out just what the little cells were doing versus what the big cells were doing. And so they separated the population and then separated
using sucrose, of all things,
by
using sucrose gradients.
by size. Yeah, so they were able to enrich for the extra small cells versus small, medium, and large. And then they could use sophisticated technologies to quantify how much DNA or what proteins were in that small cell population versus the large population.
I also like, excuse me for interrupting, I like that they use a cell sorter to determine the sizes. So this is very neatly done. It's a very nice piece of work.
And then from knowing what protein composition was of each of the populations, they could then figure out what the gene sequence was and then deduce what the gene expression profile was in each of those populations.
And by the way, they did this in great detail, right? This is done in great detail.
It is, but it's also beautifully presented. In Figure 3, they have heat maps where you can really fairly quickly scan and see how dramatically different the different populations are. And then they have what the general function is, DNA topology or transport. So
if you think about it, this is a huge amount of work because you're really comparing the proteome of two different cell types, even though they're the same, genetically the same cell. So comparing a proteome with a total proteome of a cell with that of another cell is a huge amount of work.
This is metabolomics. This is metabolomics.
Right. And it goes on and on and on. Undoubtedly, they're not done. This is going to be studied for a long time. It's going to go on and on and on. So it's a beautiful start.
Who are these people, Michael? Who did this amazing work?
This is a remarkable piece of work, and each of the authors should be congratulated. But Torben Hinsky is from the University of Greifswald, which is located in the city of Greifswald, which is located at the northwest tip of Germany on the Baltic Sea. If you're familiar with Germany, it's straight north from Berlin. So Dr. Hinsky
northeast, I think. I think it's northeast, not northwest.
Well, it's straight north. It's on the northeast tip of Germany, straight up from Berlin.
Yeah, you said northwest. And that's okay.
Oh, I'm sorry. Dr. Hinsky is a postdoc who humbles himself by simply offering that he conducts statistical analysis of omics data. But life from something as otherworldly as we can get on our planet of ours and the work that was done here, I think it's only irony that we are discussing this as our species again lands on Mars, but this time with microphones on board so we can actually listen to see if anybody's up there. But this is real. I mean, he gives us a wonderful description. He writes us a blog post on his time on the Alvin, which was the submersible that they went to collect the stuff. And he writes, and this will give the audience a color of what it's like on the Alvin. While munching on a peanut butter and jelly sandwich myself, so this is Torvin speaking, all the needs of its human occupants are thought through and provided for in the Alvin. I get to admire a huge black smoker. So this is just dumping out tons of hot, sulfur-laden debris from planet Earth through his starboard port viewport. So this extremely hot water that's coming out of the Earth's mantle is at about 300 degrees and higher. And it comes out several openings in these huge gray to black fumes. So these are hot vents in contrast to the lukewarm ones, as Elio said, the 30 degrees zone where Riftia worms settle. He tells that these worms are largely the realm of microbes, though they can't cope with the extremely hot black smoker fluids. But even at the warm vents, the microbes, invisible to the naked eye, rule the world. and this blog post goes on, and I put it into the show notes for the readers to look at, because read the blog post and then scan this paper, because it really shows you how much work these authors had to go through to get the specimens to begin to decipher this. These microbes are actually living off of the primordial soup of the planet. And they're deciphering how all of this took place. So my question, what came first, the worm or the microbe?
Ooh.
Microbe, yeah.
I mean, the worm doesn't even have a stomach.
Yeah, it doesn't.
The microbes are its stomach. So I think the microbes had to be around first.
And they're giving them the glucose, they're giving them the reduced sulfur compounds, the reduced nitrogen compounds. You know, this is Intro Micro 101. What does it take to make a cell? You need carbon, you need energy. And this hydrogen sulfide rich material is so energy intensive. This is the stuff that the early Earth was made of, and how we effectively emerge from this primordial soup is really owed to by this symbiotic grouping of microbes taking care of this tubeworm. The first time I heard of tubeworms was from Norm Pace when I was a graduate student, and Norm came for a job talk. I think it's only ironic you guys had a discussion of faculty recruiting. And so Norm was coming back to his alma mater to assume a professorship. And he's talking about his experience with these tubeworms. And this was in the early 1980s. And he asked the graduate students questions like, how does life grow at pressures of 2,500 atmospheres? How does it grow at temperatures of 300 degrees Celsius where the dielectric constant of water changes? And to poor graduate students, we're just stumped.
Who knows? So, Elio, they have to go down and get these worms, right? And bring them back to the lab. And so I noticed that these were sampled in 2014. So I guess you can't go back and say, oh, I forgot to get more. You need to work, right?
There's no store. There's no freezer on the 7th floor where you can get the supplies.
You get everything you need. You don't keep them alive in the lab, right?
No, because you don't have black smokers. Unless you have black smokers in Columbia.
No, we don't have black smokers, no.
Alvin, by the way, the submersible has a pilot who's highly trained and very specialized, and room for 2 scientists.
Yeah.
Is that all?
Good. That's all. The first time, the funny part is the first time they went down and saw the giant tubeworms, they didn't have a biologist with them. So they thought, oh, this is very interesting. They didn't know what to do with it. So next time they took a biologist along.
That's cool.
Wow.
Anyhow, it's a good story.
You know, back in TWiM number 8, we had, that was, I think, at New Orleans, ASM meeting New Orleans. We had Nicole Dubillier on, who works on these deep sea worms. And I remember she said they have no mouth, they have no anus, they have no intestine. They're just full of bacteria. I was amazed. I couldn't believe it.
They have no digestive system, whatever.
But they do scavenge the outermost large cells for food. Mm-hmm. They extract nutrients from them.
The larger bacteria become food for the worm.
Yeah.
That's right. I didn't say that.
Does anyone...
So they're not just the workhorses. Yeah. They're also the work lunch.
That's right.
They're actually accessory growth factors.
But it keeps the system in balance.
Does anything eat these worms? Do they have a predator? Do you know?
I don't know. I don't think so. They, for one thing, the outer layer of the worm is chitin, so it's not easy to chew on it.
Yeah, I just don't know what
the white stuff.
I don't think there are large predators that far down, right? Because of pressure issues.
Other than octopus. Right. No, he talks in his blog post about the octopus that is always associated with these worms. They always see an octopus with the alvin. As they near the 2 worms, there's always an octopus that goes by. And the blog post tells of a crab busting open a mussel and eating the crab. So there's got to be life there's got to be
There's
life
lots of
around
life.
these vents.
Oh,
There
yeah.
is not only there's gotta be, there is.
There is. There's not got to be. There is.
That's amazing.
That's a fantastic, weird and wonderful biology story. Thank you, Elio. Amazing.
My pleasure. My pleasure.
All right. Anything else, Michael, from the authors? Are you done with that?
Yes. I think the corresponding author, Stephanie Market, is also into. She goes out and teaches. She goes out and inspires our next generation and talks about marine life and marine microorganisms. So that's got to be absolutely fascinating as she goes out and inspires the next generation of our science teams.
All right. We have one email from Rich, who sends a link to an article, and he writes, a fascinating look into how researchers are trying and mostly succeeding to grow hard-to-culture microbes in the lab. Thanks for all you do. And he sends the search for microbial dark matter. Researchers are developing technologies to find and grow microbes that biologists have struggled to culture in the laboratory. So it's a great, it's a discussion piece with people who are doing this. And we've talked about this before on TWiM, culturing the unculturable, right?
Absolutely.
Yeah, it occurred to me as I was reading about these symbionts of the tubeworms that we've come a long way from just growing E. coli and LB at 37 on a wheel.
Yeah.
There's a whole lot more variety out there.
Indeed. All right, that's TWiM 236. You can find the show notes at microbe.tv/TWiM. You can send your questions and comments to twim@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
My pleasure. Nice to see you all again.
Where you have 6 inches of snow, right?
Yeah, it's but bright sun, so
Nice.
It's lovely.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
Where there is no snow ever, right, Elio?
Oh my God, ever. 5,000 feet. We have some snow.
Do you miss snow, Elio?
Not exactly.
Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
You have no snow either, ever, right, Michael?
We get it twice every 5 years.
I'm Vincent.
It's like I play golf twice every 5 years.
I'm Vincent Racaniello. You can find me at virology.ws. And we have snow here in the Northeast. We have about a foot or so on the ground. It's very cold, but I don't mind snow. It's very pretty, right? Especially if you get up in the morning and there's a fresh blanket, the trees are covered, you go outside and the sound is muted because of it's lovely. I'm not complaining. I really like it very much. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology. Well, Elio has left us, so let's see if he comes back. Yeah, there he is. I told you not to touch anything.
I didn't.
Okay.
No, it wasn't me. It wasn't me.
Okay.
Honest.
We have one email from Rich.